Namaste students, I am JVN Dr. Rajesh Ahmed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic to determine the blood group and what is the process of the determination of the blood group. And we know that circulatory system has about 4 to 6 liters of the blood in an adult human body. The blood has mainly two portions. First is the plasma, and the second one is the blood cells, RBCs. The plasma is mainly composed of the water, but it contains different types of proteins and other chemicals like enzymes, glucose, fat, particles, salts, hormones, antibodies, and it consists of 60% of the blood cells can be observed under microscope or by staining. The formation of blood cells occurs in bone marrow by the hematopoietic stem cells. They can be divided into three basic cell types. The first one is H cells, which are known as the blood cells, RBCs. They are colored cells and they give blood its red color. 1 ml of blood contains approximately 5 billion of the RBCs, and the proportion of the blood occupied by the red blood cells is referred to as the hematocrit and is normally 45%. So in blood samples, the experiment is we are determining which kind of blood, which blood group is in our body system. And mature RBCs are the bioconveyor nucleus have nucleus, and they do not have any kind of nucleus. RBCs are the cells which do not have any nucleus. You must know that. And they circulate in the blood system in 120 days and play important role in the oxygen supply. The next cell, which is the leukocytes, which are known as the white blood cells, and they may be of different types. These cells are key players in our immune system, and they are of different types, such as the neutrophils, lymphocytes, eosinophils, monocytes, basophils, and each of them have a variety of functions in our immunity. 1 ml of blood of an adult contains 4,000 to 11,000 leukocytes, and basically they destroy and remove the uh, aberrant cells and clear cellular debris. The next part is thrombocytes, which are known as the platelets. And the end coagulation of blood clotting process is taken care by these platelets. They act on the clotting proteins like fibrinogens, converting into the fibrin. They create a mass onto which RBCs collect and form a clot. So they help RBCs for the clotting. This prevents excessive blood loss and also checks the entry of pathogens into the body. 1 ml of blood of an adult human contains 2 lakhs to 5 lakhs platelets. So in this experiment, we need to analyze which blood samples we have. So what materials we need? First, we need some monoclonal antibody which are anti-A, anti-B, and anti-D. The blood lancet is also required. The alcohol sweat is required, toothpicks is required, sterile cotton balls are required, and clean glass slides are required, ice tray is required, and some containers may also be required. First, what we do in the process, we set up the table with all the metals which are required. Okay, we collect all the samples and place the monoclonal antibody kit on the ice tray. It is required so that they can be in the proper temperature conditions. Then we start the experiment. First, we take a cotton and we take alcohol swab and rub in the area of the blood. Suppose the blood should be collected from here, so we rub in this area, and with a sterile lancet, we put some pressure on the tip of the finger from where the blood will be sampled, and we prick the finger with the lancet, and as the blood start bleeding, we make one fall of the blood from the glass slide. So one, two, three drops are made on the glass slide. One, two, three. One, two, three. Okay, and then we place a cotton block on the side where the blood was pricked, and we using the thumb, we put some pressure on the area of the blood from where the blood was collected. So we put the cotton swab here, and we discard the cotton swab. After that, the blood has been collected. We have placed the blood drops on the slides. Then we take NTA, which is the white bottle NTA, and we suspend the content. We suspend the content of the NTA, and we place one drop of NTA on the first sample of the blood. Okay, and then we mix it. Then we take some NTB, and we add one drop of NTB in the second number of blood drop, second spot of the blood, and use the NTB again. Then we take the NTD, which is colorless one, and we take one drop of NTD on the third spot of the blood. Then we take a toothpick and mix all the content very well using a toothpick, and then we discard the toothpick after using it, and then after mixing we wait for a few seconds or few minutes for the observation of the results, and we can observe the results by that. For the analysis of the results, when we go to analysis of the results, we must know that what blood group can be possible: blood group A, blood group B, blood group AB, and the blood group O. So these are the possible blood groups. And what are the content by which we identify the blood? So in the blood group A and B, AB and O, there are some antigens and some antibodies. Okay, in the blood group A, the antigen is antigen A. Okay, in the blood group, the antigen is antigen A. In the blood group, the antigen is antigen B. In the blood group AB, antigen A and B, and in the blood group O, and no antigen is found. Similarly, if we talk about the antibodies in the blood group A, antibody B, NTB is found. Okay. In the human with blood group B, anti-A antibody is found. In the blood group AB, there is not any kind of antibody. And in the blood group O, anti-A and anti-B is found. So the blood group is identified by its antigen, means antigen blood group A has antigen A and it has antibody B. The blood group B has the antigen B and it has antibody A. Opposite. The blood group AB has an antigen A and antigen B, but it do not have any kind of antibody. So that's why it is known as the universal recipient, because it do not have any antibody. Similarly, the blood group O do not have any antigen. That's why it is known as the universal donor, and it has anti-A and anti-B. So how the results can be observed? Uh, by using this slide, we can analyze how we can analyze our blood. So in this uh, experiment, these are the normal blood, which is here, and there are 4 slides in which the blood has been added. 3 drops, 3 drops, 3 drops, 3 drops of different blood groups. Suppose this is the spot number 1, spot number 2, spot number 3. Suppose there is a agglutinating clotting in the spot number 1, which indicates that the blood has anti-A and antigen A. So antigen A and anti-A has been reacted to each other, which indicate that the blood is A. And this is for the analysis of the RH vector. If it is positive, then it indicates that the individual is RH positive. So the 2 spots are showing clotting here. So it indicates that the blood group is A positive. In the second type of blood group, if we observe that there is not any kind of clotting here in the first spot, there is a spot in the second spot when which indicate that after adding of the anti-B, there is a clotting, and similarly the clotting is also found in the third spot of the blood, which indicate that after adding anti-B, the blood is clotted, which indicate the blood group is B and positive, B positive. Then in the so third slide here, if there is any kind of clotting in the first spot, we are finding that there is a spot, there is a spot, there is a clotting in the first spot. In the second, the clotting is found, and in the third spot, the clotting is also found, which indicate the blood group is A positive, it is also B positive, and it is also positive, RS positive. So AB positive, the blood group will be AB positive. And in the last blood type, we are observing here that there is not any spot in the first, there is not any clotting in the first spot, there is not any kind of clotting in the second spot, and there is no any clotting in the third spot. So no spot, no spot, and no spot means the blood group is negative, and it is it has no impact on the anti A and anti B. So the blood group will be O negative, where not any kind of clotting is found. So I hope students will have understood that what is the basic of the identification of the blood group. So what students have learned about from this lecture, students will be able to understand, explain the principle and procedure of the blood grouping, explain the blood grouping experiment, and what is the complete experiment on the blood grouping. Students have also understood that what kind of antigen and antibodies is found in different kinds of blood groups. Similarly, some another things can also be understand that what kind of cells are found in the blood. So my lecture is over here. I hope the students have been better understood. Thank you very much.